Welcome to this Labroots podcast, The Power of Attraction, exploring Dynabeads in sample preparation. This podcast is brought to you by Thermo Fisher Scientific. Partner with our global team of experts to navigate and conquer challenges from discovery to commercialization with fast, reliable, and cost-effective solutions. To find out more, go to thermofisher.com/Dynabeads. Now here is your host for this podcast, Senior Product Manager of Dynabeads Products, Claire Mount Lund.
So welcome, podcast listeners, to our podcast series, The Power of Attraction, exploring Dynabeads in sample preparation, your window into the world of scientific research. So today's episode, we are going to be talking about the really exciting area of liquid biopsy, a topic really close to my heart. So I'm really excited about this one. And we are very lucky to be joined by Marc, an expert from Thermo Fisher Scientific, a world leader in serving science. Before we jump in, just a reminder to our listeners that the products and also the techniques that we'll talk about in this podcast, they are for research use only purposes and not for diagnostic purposes. And with that, Marc and I are going to spend the rest of our time here exploring the innovative role of Dynabeads, magnetic beads in liquid biopsy. And what better place to start than if our guest could introduce himself for us, the listeners.
All right, my name is Marc McGowan. I'm a scientist here at Thermo Fisher. My area is with cells, cell biology, immunology, and everything in between.
Perfect. Sounds like a perfect fit for this particular podcast. So welcome, Marc. I'm going to start us right back to the basics, if I can. If you could just give us kind of a brief introduction to the current landscape for cancer and cancer diagnostic, cancer research, those kind of areas.
Yeah, so I mean, cancer is still one of the world's leading causes of death, and statistics state and show that 1 in 5 people will be diagnosed with cancer in their lifetime, which is still really high, regardless to all of the advances we've been making since 1959 and onwards. So really the fundamental of that is the detection, catching it early. You can go anywhere and look at the news and ask anybody, and a lot of them will turn around and say it's detection which is the key to success.
Yeah.
And that is what we're focused on here. In our product ranges, especially using the Dynabeads magnetic beads as our go-to for getting that early capture and enabling researchers to move forward.
And sounds like a really powerful area to be focusing on. Are there particular areas that researchers are focusing on in order to raise the bar on the way in which we can detect cancer?
Yeah, I mean, the way that things were done, if we go back to actually detecting it and actually pulling something out that the researchers can use as a starting material, we're talking about the biopsies. We're talking about collecting of cells. Fundamentally, what people are trying to do now and are doing now is using less invasive techniques to take a biopsy. And because what was commonly done and still is in some areas, but not so much in research, is that a primary tumor biopsy will be taken. And that's really, for researchers, that's going to be very, very difficult to obtain unless you're in the right circles. And it's not nice for the patient. So as an alternative to that, well, not really an alternative, but sort of like a neighboring biopsy is the liquid biopsy. This is a blood test. This is a sample of urine. And from within those samples, you can take out biomarkers. You can isolate tumor cells from there. There's a whole host of things you can isolate. So really, it's all starting with the material. And taking a liquid biopsy can give you a lot of information.
Exciting stuff. And if we can stay with that topic of the liquid biopsy, definitely sounds like there's some advantages there. is there more that you can explain to us around liquid biopsy? You mentioned a few things that you can detect in there, the types of samples you can use.
Yeah. So, I mean, we talk about it, and you get let's say you get a blood sample, for example, and a blood sample, you can separate it into different phases, and from in those phases, you can isolate various biomarkers. Particularly, you can isolate the circulating tumor cell, which is it's a very, very big and a hot topic, because again, from that phase is not just the tumor cells, but you can also get immune cells as well. Giant topic. I'm sure I can chat a bit more about that later on. And you can also isolate some of these what we call extracellular vesicles, particularly exosomes, which has been around for a long time, but has now come back because people are detecting and finding out that they can learn so much from these exosomes, which are secreted by a lot of cells, many cells in the body. And with these extra vesicles, again, I can go into more detail later on, you can generally find its cell of origin and also biomarkers, and a particularly cancer cell, you can find different biomarkers within there.
Fantastic. It sounds like we have a lot of things still to dig into here. Before we go into exploring those biomarkers in a bit more detail, the samples that you get from a liquid biopsy, can you describe anything around those and any particular things that you have to pay attention to about the samples? Like, I'm thinking, are the targets abundant in there? Are there a lot of them? Or
Yeah. So no, what you're taking from a sample is you're taking a lot of different cell types, a lot of microRNAs, a lot of proteins. It's a clustered. It's a cluster. So what you want to do is be able to isolate what you're actually looking for, and it is the proverbial needle in a haystack. Except the needle that you're after is slowly eroding away, so that getting that to that needle quickly, as efficiently as possible, without damaging it. That's the key to research and early detection as well, which researchers are still, I mean, just go in the publications and that's what you'll find. And that's where our technology comes in. The Dynabeads magnetic beads come right into this. Because what we can do, again, fundamentally, we can go into the haystack with a large magnet, and which the Dynabeads are, they're magnetic, and we can pull that out specifically, or we can reduce the size of the haystack by removing all the clutter to find what we're after.
Yeah, definitely sounds like a bit of the theme I'm getting in this podcast series. There's definitely a lot of versatility in the way that Dynabeads can be used, and definitely sounds like this area really needs that good sample prep.
Yeah, there's a lot of plasticity in it.
Yeah. Good. So can you tell us a little bit more about these magnetic beads isolation? Can you take us through a little bit of detail about how you can actually use them in this setting?
Yeah, so if I was just to bring an example, let's say we wanted to isolate a T cell, for example, to put that case in point. Now, the T cell is full of biomarkers on the surface as well as intercellular, but it's the surface that you want to challenge. And we challenge that with one of these Dynabeads, and we can put on there an antibody that specifically detects that T cell, for example. So that will go in to the blood sample, to the biopsy material, and it will pull out that particular T cell that you're interested in. I mean, that's just a one-to-one basic. That's just to give you an idea. And that is one way of doing it. Again, that doesn't just stay to there. We go to circulating tumor cells, where we will put what we refer to as EpCAM, as an epithelial cell adhesion molecule, which will be present on the tumor cell. And again, as it's free floating around the circulatory system, you can, in a blood sample, you can go in there and specifically isolate that cell using the antibodies that are coupled to the ends of these Dynabeads onto the surface. So that is one way of doing that.
Interesting. Definitely can see the analogy to the needle in the haystack, going fishing for what exactly you want.
But again, the the emphasis on that is to be quick, is to isolate the cell, because once the cells are outside of the body or even outside of a cell culture, these cells are not happy. And it sounds interesting when you talk about tumor cells, but they are still fragile cells, and you want to keep them intact for whatever reason that you want. You want to culture them, or you want to take them straight for analysis. You want them as in their best healthiest state you can have them in, and getting them out quickly, efficiently, without damaging them. Dynabeads do that.
Perfect. Good. And you talked a little bit about it already, but if we could we dig a little bit deeper onto the exosomes? I'm more familiar with the cells, but the exosomes, are they the kind of similar kind of thing to the cells, that they have biomarkers on them that we can use? Or tell us more, please.
Yes, I'll do my best. Do my best. Yes, so exosomes, these are part of the extracellular vesicles, and they come in different sizes. Not the exosomes, but the extracellular vesicles become in different sizes.
Right.
But for this example, we'll look at exosomes. Now, these are secreted. They're kind of like, if you can imagine, like budding. So these are released from the cells, but when they're released from the cell, it used to just believe that these were just the waste of the cell, until researchers got a little bit interesting, and one of them asked the question, or maybe a few of them asked the question, what are they? That's how it starts in science. These have been found to carry a lot of these are cargoes for a lot of the intracellular networks and proteins, mRNA, DNA. Right. These are released from the cells, along with some of the surface biomarkers as well. Now, are you familiar with these online gene family trees? Where you give a blood sample, and then somebody would take it and do something interesting with it, and then they will say, oh, this is your family tree. It's often the case, has many branches. These exosomes behave in a similar manner. In the terms of, if you think of a family tree, you can look at this, you can pull this exosome, release it from a blood sample or a urine sample, and you can look for biomarkers on that, and you can trace it back to where the cell of origin, because of the biomarkers, or at least cut down the field to say, well, it's probably something like this. And I mean, I can go into the problems with getting these exosomes, because it's all right saying, we'll get these exosomes out, we can see the biomarkers, and we get all these wonderful information that we can trace back. But again, it all goes back down to the starting material, and what people are currently using, and not so much currently, but pretty much I call it an outdated method, because of the way that they get the starting material. That it would, in this case, you've got your sample, you have your blood sample, you move it on, then you want to get the exosomes out of there because you're interested in everything that's inside, all the gubbins inside it, or something, And the way that people have been doing it before is this thing called ultracentrifugation. I mean, you put ultra in front of anything, it's usually not that a good thing. So this is this method has been around decades, and decades, and it really is. I mean, if you're talking about a method that's decades, I mean, you wouldn't use technology that's decades old. It doesn't work like that. It's obsolete. And the reason why we're saying, or I'm saying, it's really not a good option, is because every time you come to spin these samples at very high velocity, you pellet them, and you have to wash them. And every time you wash, you lose exosomes, because these are very small, which requires an incredible amount of G-force to get them down and to keep them in the one place. They're not like cells. So every time you do that, you're losing exosomes. You're not even too sure that you've got exosomes at the end of it. And it's a long process, and ultra anything can be quite dangerous. And the alternative and the go-to for this is something really quick, and that is our Dynabeads again. Either you can pull them out specifically and release them, and that's done very quickly without centrifuging. So you get a lot more.
How quickly are we talking?
It could be anything from 15 minutes
Impressive.
You know, so I mean, for example, the SACS virus beads that we have rely on an ion exchange, and these exosomes are negatively charged. They're attracted and can be released as well. So you will get exosomes very, very quickly.
Yeah. Sounds very simple in a way. So yeah, and I think you already kind of touched on. It sounds like these types of liquid biopsy samples are quite precious samples in terms of like there's an abundance of these the targets that you're looking for. So it makes a lot of sense that you don't want to lose things along the way.
No, you don't want to lose anything along the way. It could be quite detrimental.
Makes sense. Good. So it sounds a bit more back to the kind of needle in a haystack thing that Dynabeads are doing again here.
Yeah, I mean I think it's a perfect analogy because needles are metal and Dynabeads are magnetic. And,
Good point.
Bob's your uncle.
Yeah. Good. Very impressive. And we touched on the exosomes. Can we also then go to the circulating tumor cells? Is there more you can tell us there about how we want to isolate them, how Dynabeads can help?
Yeah, so again, if I bring the needle in the haystack analogy back, the circulating tumor cells. So in the biology of things, you have, if a tumor will generally originate in one area. We're not talking about cancer now, so that's a little bit slightly different. Well, it's different, but it's still not good. Well, neither of them are good, but the tumor is often a mass. And when this becomes detectable, often it's quite pronounced at that stage. And what we're always talking, and researchers are always talking about early detection. So when you have this tumor cell, some of the cells will end up breaking apart through any means. It could be metastasis, it could be a whole list of reasons. The cells just come apart and they enter the circulatory system.
Right.
So they can spend any time from a few weeks to a few years just going around the circulatory system. It's those that are important because those are what researchers are after. To be able to capture this cell to either culture it out or do downstream processes. So they want this cell in a mint condition as possible. And to do that, you will have these Dynabeads that we've got. Again, labeled with something specific, an antibody against them. In this case, it's EpCAM.
Yeah, you mentioned the EpCAM.
So it will bind to the tumor cell, the circulating tumor cell. So this will just be one cell going around. For the talk, one cell. There are very, very few. So when we go back to precious sample, we're talking about, you can't just go back to the freezer and pull out another one if something doesn't work. It has to work there and then. If there's a tumor cell in there, you will pull it out with the Dynabeads because it's going in there specifically looking for it and capturing that and then just magnetically withdrawing it from the blood and
there you have
capturing it in perfect condition. Again, but a lot of researchers might want to do something with that. They want to, we've got this personalized medicine, and that's into research as well. So if you've got a lot of samples, or from a blood sample, and you want to look at different compounds or test something about these cells, and you want to grow them out, but you don't want the surface touched. Because that might cause activations if it's looking at certain pathways. I mean, it shouldn't, but sometimes researchers are quite clear that they don't want it touched. Then you can use the removing of the haystack and then pull it out that way. And having it untouched and then looking at particular pathways inside the cell. So that goes back again, precious sample. Let's say if you try and centrifuge these out, you're going to keep damaging the cell. And often, what if there's just one cell and you're washing and of course, you don't do that, but in this case, there it goes, down the drain, never to be seen again. You've lost it.
Interesting. So CTCs, we've got those covered. Are there any other cell types that we can think of that have some application to cancer that could also be interesting to isolate?
I mean, immunology. I mean, we go immunotherapies, immuno. That's taken off hugely, and it's taken off here as well in Norway.
Research into this area,
I mean, thanks to Ursula Helland Radium Hospital really made a big thing about this, and quite rightly so. Why is immunotherapy? Why is immune system very, very interesting in cancer research? Well, let's go back to a little bit of the biology. When we talk about the immune system in tumors, in cancers, we'll often be referring to them as a hot or a cold tumor. Okay, so when we say a hot tumor, it means there's inflammation. And a lot of people, we know what inflammation is. It's when you can't get out of the chair, your knee hurts, you've a whole host of reasons. It's not associated with good things.
No.
However, in cancers and tumors, it is. And that's what's interesting. We want to know, why is this a good thing? What's happening to those immune cells? The Ts, the Bs, the Tregs, and everything, and NK cells as well. Then you've got the other end of the spectrum, which is the cold tumor. This is when there is no inflammation. And this is of interest. Why aren't the immune cells in there?
Yeah.
Why is there no immune response? We know a lot more now, but there's still things to learn.
Yeah.
I mean, so what we can do again with our Dynabeads, we just got another antibody in there to go in to look at each specific immune cell that would be in the area. And you can pull those out and say, well, why is this not active?
Yeah.
What's causing it to be?
Yeah.
And you can pull out some of the tumor cells as well. What are they doing to deactivate this or to cloak it?
The links between
Because in cell biology and immunology, we've referred to it as when it goes for detecting the immunosurveillance. So we'll go over and have a look. You know, I mean, my analogy is, think of anything from Star Wars, trying to find the rebels, and it's going around, the little heads are bobbing down, can't see them in the snow, and that's the immunosurveillance.
Right.
And then what we can do, we can just take that out, take solo out, so we can have a look at them, see why.
Try and understand it.
Yeah.
Can never get tired of Star Wars analogies. So
Neither
thank
can
you
I.
for that.
Neither can I.
Very good. I mean, this is just incredible, the number of ways in which we can study all the cancer different.
I mean, this is technology that's here today. I mean, who knows what's going to come in from the future as well.
Totally. Yeah, we've learned a lot. It's so much we can get. Any other things that you're thinking are coming ahead in the future in this space where our Dynabeads can also be?
Well, I think, if I go back to the immunosurveillance and the cold tumor, we have this bead that can activate T cells. So again, it's the interesting thing is not only if you want to grow T cells for whatever reason you want to expand them, we have a bead called a CD3, CD28 bead, which activates the T cells. Anything, pretty much all of them. Now, if you take something out of a cold tumor, or you have, you can mimic that, you can actually see, can this be reactivated?
Okay.
I mean, you think about that powerful research tool.
Yeah.
If a researcher was to publish or early stage research in a company and say, we can reactivate them and we know how. I think how powerful that's going to be in oncology, in immunotherapies.
Yeah. Wow. Really staggering. Yeah. A lot still to come, I can see. But as you've already talked about earlier in this podcast, there are some constraints around samples. There's also some constraints around our time. And sadly for us, our time on this podcast has come to an end. But I have learned a lot, and I'm super grateful that you could join us and share some of your insights in this really, I think, really important and exciting field of liquid biopsy. So thank you so much for joining us, Marc.
No problems.
And thank you, listeners, for joining us again. I hope you've learned a lot. And if you're still keen to learn more, don't forget that we do have a back catalogue of podcasts, and you can learn about, for example, the origins of a Dynabeads technology and all the history surrounding it. And also we delved into the space of proteomics and immunoprecipitation. So be sure to look out for those episodes as well. But for now, thank you again for joining us today.